My name is Qingfeng Wu. I graduated from Fudan University for my bachelor's degree, and then I moved to the Institute of Neuroscience, Chinese Academy of Sciences, based in Shanghai for my PhD degree. After graduating from the Institute of Neuroscience in Chinese Academy of Sciences, I moved to the Johns Hopkins University for my postdoc training, and then started my own lab in 2017 in the Institute of Genetics and Developmental Biology, Chinese Academy of Sciences, based in Beijing right now. Next, I would like to introduce my research in the lab, and I think our lab focused on the hypothalamus development, function, and related tumors. And I think my current research is quite different from my previous research training. And I think why I focus on the hypothalamus is just because the hypothalamus controls many important physiological functions, including the feeding behavior, sleeping, and the reproductive behavior, and so on. And basically, I think the hypothalamus can control the endocrine system, autonomic nervous system, and regulate the physiological homeostasis and many native behaviors. So I think given the importance of the hypothalamus, and I think it's very, very fundamentally important to dissect the development and function and related disease of the hypothalamus. I think this paper is based on our previous research about the cascade diversified models of the neuronal diversification in the hypothalamus. And I think the neuronal diversity in the hypothalamus is super complex. It contains just more than 100 types of the neurons. So the neuronal diversification is highly complex. So we previously proposed a cascade diversified model, but we think that it may not underline such a supreme complexity of neuronal production in the hypothalamus. So in the current research, we try to understand the development and the evolution of the mammalian hypothalamus. We collected just more than 350,000 cells from the human, macaque, and mouse hypothalamus across different developmental stages. And I think currently we developed a very, very big website to show the atlas of the developing hypothalamus across different species. And the key findings in this work include we found that the neural patterning and the neurogenic lineages are basically conserved across different species. So for the neural patterning, we found that we proposed two very important concepts. The first is the tertiary organizers, and tertiary organizers can release many different morphogenes, including sonic hedgehog, Wnt, and so on. This is serve as the extrinsic cues for the regionalization of the developing hypothalamus. And the second, we also found that different genes in the Fox gene families can segment the developing hypothalamus along the anterior hypothalamus axis. And I think this is very, very interesting findings. But for the neurogenic lineages, we realized that the cascade diversified model may not sufficiently underline the generation of such a complex neuronal diversities. So we think that the hypothalamus may be composed of different sublineages. So we indeed identified a few different progenitor domains, and each of the progenitor domains can give rise to distinct sublineage. So we construct a very, very complex hypothalamic cell lineage during the development. So we believe that the neural patterning and neurogenic lineages are conserved across different species. But also we identified a few very interesting innovations in human hypothalamus. The first, we found a very specific types of the neurons which are highly enriched in the human developing hypothalamus. And second, we also found that neuromodulator expression are highly expressed in the human hypothalamic neurons, including the ion channels, receptors, and the ligands. And the third, we found that two distinct neuroendocrine neurons, including the GNRH and GHRH neurons, are highly dispersed within the human hypothalamus. So they have more widespread dispersion than in the mouse developing hypothalamus. And lastly, we identified a few interesting hypothalamic dopaminergic neurons. And in these neurons, the human dopaminergic neurons, they express the more excitatory transmitters, glutamate, which are coupled with the dopamine. But in the mouse developing hypothalamus, they are more coupled with the GABAergic neurotransmitters. And also we found that some of the neuropeptides are reshuffled within the hypothalamic dopaminergic neurons across different species. And these are 4 important innovations we identified in human developing hypothalamus. Yeah, I think this is a very interesting experience. So this is my first time to submit our manuscript to multiple journal submission system. So I think the most compelling point is that this system allows us to receive the feedback from multiple journals at the same time. And I think they can provide us the tiered revision recommendations based on each journal's specific standards, allowing the authors to respond strategically at their preferred levels to meet the editorial expectations. So I think the only limitation of this system is that the initial review may take a little bit longer than expected due to the coordination among editors. However, I think the workflow becomes highly efficient once a target journal is selected. The editors transfer that manuscript directly to that journal's team, ensuring the faster handling and decision making. So I think it could provide us more flexibility to respond to different journals' specific standards. Yeah, I think we just choose the cell, developmental cell, and neuron. So and lastly, our manuscript was accepted by the developmental cell. Yeah, sure. I think recently we submit another paper to Cell Press, but in the future, I think we will have more papers submitted to the multiple journal submission system. Yeah.